Good day, everyone. So here at Magic School, we'll be continuing our enzymology course, and we're considering active sites and zymogenes in this video. So yeah, let's get into it. So yeah, the active site is the part of an enzyme or the portion of an enzyme where the substrate binds to. The substrate doesn't bind to the entire enzyme but binds to a particular portion of the enzyme, which is the active site. And then the active site confers on the enzyme its specificity to the particular substrate it can bind to. So now the actually characteristics of this active site. Now it is hydrophobic in nature. The active site creates a non-polar environment for this substrate. Water molecules are found there only when probably is a water molecule that is being reacted, or probably water molecule is needed for the reaction. It is flexible. We see under the methods of substrate binding to an enzyme, we have the lock and key, and then we have the induced fit. Under the induced fit, we see that the active site can be modified or modifies itself to the substrate to be able to bind to the substrate. Now it is also reactive, and then it contains special residue that are responsible for the actual binding to the substrate. Now we know that the enzyme is made up of amino acids. So special amino acids that are placed at the active site enable or promote the binding to the substrate. Now it binds to the substrate by non-covalent interactions. So now these amino acids in the active site that bind to the substrate, they bind by non-covalent interactions such as hydrophobic interactions. And now because of their non-covalent interaction, the binding of the substrate to the active site is reversible. So after the substrate has been converted to its product, it can easily be released from the active site. Now also the shape of the active site is complementary to the substrate. It has a shape that fits a particular substrate, and then the active site is small relative to the size of the enzyme. Relative to the entire size of the enzyme, the active site is quite small. Yeah, so those are the characteristics of an active site. Now zymogenes are proteins that are synthesized in the inactive form. They are proteins because these zymogenes could be enzymes or could be hormones or proteins in general. Enzymes are proteins, and when they are synthesized in their inactive form, they are called proenzymes. Now, for these enzymes to become activated or be able to carry out their work in the body system or to carry out their particular function, they need to be activated. And then this activation can be done by proteolytic cleavage by protease, and the second one is autocatalysis. Now, for the first one, proteolytic cleavage by protease, I'm giving an example of a hormone, insulin. Now, insulin is secreted in its inactive form. So now it needs to be activated, and is activated by protease for it to be able to carry out its function in the body system. And then the second one is autocatalysis. Now pepsin is secreted in the body as pepsinogen. Now this pepsinogen needs to be activated to pepsin to carry out its work in the body. Pepsinogen, it is activated at a pH of around 2 to 3, and then because of the HCl in the stomach, the HCl brings the pH of the stomach to about 2 or 3. So now when the pepsinogen gets to the stomach, it is activated as a result of the low pH of the stomach, and is able to carry out its activity, or able to carry out its function of breaking out peptide bonds and breaking down proteins. So now let's look at some examples of these zymogenes that cut across both enzymes, hormones, and just some proteins. So now we see trypsinogen. Most digestive enzymes are synthesized as zymogenes, because where they are being produced is not their site of action. They need to be transported for them to carry the activity. So where they are synthesized in an inactive form, so they don't carry out digestive function on the surrounding of where it is being synthesized. So now we have trypsinogen, pepsinogen, chemotrypsinogen, and pepsinogen. They are actually digestive enzymes, and then they are secreted in their inactive form, and then they are made active or they are activated when they get to where they will carry out their function. And then I've given an example of insulin. The inactive form is proinsulin, and then it's converted to insulin. And then we have procollagen being converted to collagen, plasminogen to plasmin, and all that. Then for proplaestylase is also kind of a digestive enzyme that helps to break down fats, proteins, and carbohydrates. Now angiotensin which is the active form of angiotensinogen. It's an hormone that regulates blood pressure and the fluid balance in the body system. Now, plasminogen. I mentioned plasminogen, the inactive form of plasmin. Now, this is a wound clotting factor or a blood clotting factor. So that will be all for this video. Thank you, and don't forget to like and subscribe. Thank you.